Okay, so let's talk about the SLIM. So SLIM, it stands for Site-Directed Ligase-Independent Mutagenesis. And it's a simple and efficient way to introduce mutations into a plasmid DNA sequence. The plasmids will then get introduced into cells that will divide and clone more copies of itself and the plasmids inside of them. So here's the procedure. So step 1 is primer design. In SLIM, there are 4 primers to be used. So we have 2 normal forward and reverse primers that will, of course, bind to the sequence and trigger DNA synthesis. And we're going to name them F short and R short. The other primers are quite similar to my FS and RS, except on the end, as you can see here, they have a 5' adapter sequence attached as a tail. And these adapter sequences, they will be the key to introducing mutations into my plasmid sequence. We're going to aptly name them F-tail and R-tail. Note that these adapter sequences are complementary in the forward and reverse. Step 2 is PCR. So here is my plasmid with my gene of interest. And what we're going to do first is we're going to heat up the solution to break the hydrogen bonds between the DNA in the denaturation process. Then the primers inside the solution can anneal to the DNA, attach themselves onto it. They will then extend in divergent directions to create a linear PCR product, like this one. So of course, in this step, I used FT and RS. These are the 2 primers I used. I can repeat this particular step with different primers, FS and RT. And I'll create this particular PCR product. Of course, these both strands I've created are complementary and are currently bonded to each other. Now what we're going to do is we're going to mix them together. So what we're going to get is in the solution, we're going to have this particular product, and my original plasmid, And I don't really want my original plasmid, right? I only want these 2 products going on in my solution. So that's where step 3 comes in. We're going to add DNP1. And the DNP1, it will digest the original template plasmid, which then of course allows me to remove it from the solution. And now I have a solution which only contains these 2 PCR products we made earlier over here. So what we're going to do to these PCR products is that they are going to undergo denaturation and reannealing, our step 4. So essentially what's happening here, we're heating to separate the sequences, and then we're going to cool the solution down so that the sequences can form new bonds with each other. You can think of it as taking apart Lego blocks and then putting them back together in different ways. And we're going to do this step multiple times. Of course, there's going to be many different double-stranded species that will get generated from this step. But here are the 2 species we want in particular. I'm going to focus on this one because they're quite similar. So I want to direct your attention to these complementary overhangs. As we remember earlier when I was talking about primers, the adapter sequences are complementary to each other. So what's going to happen is these complementary overhangs can kind of act as sticky ends that will join the DNA into a circle in a process called spontaneous circularization, like this. And now what I get is I get a lovely plasmid that's been mutated. So this plasmid will then get transformed into E. coli, which can create colonies on selective media. And you might think to yourself, oh, but what about, you said that we create many different combinations of these strands. What about the other strands? Well, we don't have to worry about them because the other strands are linear strands. Only these 2 particular products will actually undergo spontaneous circularization to form a circular plasmid. And well, only circular DNA molecules can be transformed by E. coli. So we can ensure that only this will be in our product. One of the great benefits of SLIM is that it's quite simple, as we can see from these steps, and it has a very high efficiency compared to the T4 ligase cloning we did in the labs. SLIM also doesn't use ligases or require restriction enzyme sites in a gene of interest, which allows for more precision and flexibility. One of the uses we can have for SLIM is that T4 ligase cloning. It's typically used to insert larger DNA fragments, and it's a bit less optimized for making small edits. And SLIM can fill that gap and fine-tune some of the mutations we might want to add to a plasmid. So we can use SLIM as a complementary tool in this way. And that's just one of the methods we can use SLIM in the lab.